Dear colleagues, welcome everybody to this new seminar of the eSchool series, which are organized by the European Society for Gene and Cell Therapy. My name is Juan Bueren. I am the current Vice President of the ESGCT, and it is my honor today to introduce to you to a great scientist and also a good friend, my colleague Zoltan Ivics, who is the head of the Medical Biotechnology Division at the Paul Ehrlich Institute in Langen, in Germany. Zoltan Ivics received his PhD in molecular biology in 1994, and after his postdoctoral studies at the University of Minnesota and in the Netherlands Cancer Institute, he was appointed as a research group leader at the Max Planck Delbrück Center for Molecular Medicine in Berlin, and thereafter he was appointed as head of division of the Paul Ehrlich in 2011. Professor Ivics' major achievements is the molecular reconstruction of the Sleeping Beauty transposon, and also the development of technologies based on Sleeping Beauty gene transfer for a wide array of applications involving genetic engineering. He has published more than 130 papers in peer-reviewed journals and is a co-inventor of 12 patents, many of them related with Sleeping Beauty technologies. He has received the recognition of the Molecule of the Year in 2009 for developing hyperactive Sleeping Beauty transposase, which has opened the door for several clinical applications. Some of them will be presented today. He is a current member of the board of the European Society for Gene and Cell Therapy, is member of the board of the German Society for Gene Therapy, and also member of the Committee for Clinical Trials and Regulatory Affairs of the German Stem Cell Network. And he is an elected member of the Academia Europaea since 2018. In his today's presentation, he will provide an overview of the Sleeping Beauty Transposon technology, and he will highlight the most recent developments of the system with a particular emphasis on clinical applications. Well, as in other seminars, you are invited to send your questions during the seminar, and I will try to raise them at the end of the seminar. Finally, also, I would like to announce that after this meeting at 3 o'clock, there will be another one that will be given by Dr. Amy Patel from the Wistar Institute in Philadelphia about novel vaccine strategies targeting SARS-CoV-2. So thank you very much. And now, Zoltan, it's time for your presentation.
Thank you very much, Juan, for this very kind introduction, and greetings to everyone out there. It's my privilege today to tell you a little bit about the history of this intriguing gene delivery system and kind of give you a brief update on the most exciting applications. This is, of course, a very personal view of mine. So I will be talking about the Sleeping Beauty transposon, a molecular parasite tamed for genome engineering. And let's get started with this. Let me just kind of summarize or give you a summary slide on where we are positioning this transposon-based tool on the map of gene delivery vector systems or technologies altogether. Roughly speaking, we differentiate non-viral and viral vector systems. The difference is obvious between them. Viruses have an active mechanism to transduce or infect a cell and deliver the genetic material into that cell, whereas non-viral systems is kind of a collective term that covers all different kinds of technologies that are not based on viruses. Even without within viral systems, there are so-called integrating viruses that permanently integrate a transgene into the genome, whereas other types of vectors, including AAVs and other types of vectors, adenovirus or baculovirus, are non-integrating system. Now, the point of this slide really is to kind of highlight the transposons, and Sleeping Beauty is one particular transposon that I will be concentrating on today, is this particular feature that kind of links these two different worlds. It's kind of a crossroad between non-viral and viral technologies. Why do I say that? The reason for this is that a Sleeping Beauty transposon-based vector is non-viral, typically speaking. It's maintained as naked nucleic acid in the form of classical plasmids or other nucleic acids, but it's kind of making a link over to the viral systems by having an inherent capacity of stable genetic integration. That's what transposons do. They can integrate their own genome into chromosomes. So it's kind of an interesting combination of these two worlds that is linked by a classical transposon system. So let me now quickly go through the major and basic mechanism of the transposition process. We also call it cut and paste DNA transposition. And in a minute, you will see why we use that terminology. So first of all, the transposon is sitting somewhere under natural circumstances. Right now, I'm not talking about a vector. I'm talking about the transposon that is a component of a genome out there in nature. So the transposon is composed of two major functional units, a DNA unit highlighted here by these black arrows on the left and on the right. These are called terminal inverted repeats that kind of define the boundaries of this jumping genetic element. And the second component is a coding region for an enzyme that is called the transposase. So this transposon is sitting somewhere in the genome. I highlighted here this particular locus as a donor locus or donor chromosome. As a very first step of the transposition reaction, the transposase gene will be expressed, producing this protein or enzymatic factor, which is called the transposase. This is required for the transposition reaction. As a second step, this transposase polypeptide, once it's expressed, will sit down on these terminal inverted repeat sequences because the transposase is a DNA binding protein that is specific for these sequences. And following this initial binding reaction, the transposase will catalyze two separate biochemical reactions. Collectively, we use the terminology cut and paste DNA transposition. So the first step is excision. This means that this transposon is physically speaking cut out from the donor chromosome. That's number one. And number two, this physically excised or removed transposable element unit will integrate into another chromosome, which is the recipient chromosome. So this is the natural way of DNA transposition. It's kind of a translocation process that is mediated by a DNA recombination mechanism dictated by a DNA component and a protein component. How do we tame this natural DNA integration reaction in the context of genetic engineering? This is highlighted on the bottom of this slide. So practically speaking, what we can do is that we can replace the coding region for the transposase with any other gene of interest or any other piece of DNA, thereby creating an artificial transposon. And this artificial transposon is typically maintained in the form of a plasmid, a classical plasmid that we can propagate in E. coli cells. This is why we call a transposon vector a non-viral vector, because the typical format of this is a straight plasmid. But the rest of the reaction is exactly the same. cut and paste transposition can take place out of this non-viral vector. Transposase is provided in trans, and I will highlight during this presentation in what way we can express the transposase in target cells. Transposase will sit down on the terminal inverted repeats and catalyze excision of the transposon out of the plasmid and permanent integration into a recipient chromosome. So this is roughly speaking, the basics of the major biochemical steps that are involved in DNA transposition. And this is the exact basis of using these transposons as tools for genetic engineering. I will cover a few aspects of applications of Sleeping Beauty technology in vertebrate cell engineering, just to go through a few areas of application, kind of just as a bullet point list. We can use Sleeping Beauty vectors to engineer cells in tissue culture. We can use the transposable element vector system to introduce transgene vectors into the germline of animals. This is the area of biomedical research that is based on animal models, either knockout or knock-in animal models, and I will show a few examples for this. Functional genomics, I will not cover this at all in this particular presentation, but this is also a very exciting area of applications where we actually allow the transposon to kind of randomly hop into gene sequences, thereby leading to certain phenotypes that can be annotated in functional genomic screens. And finally, gene therapy or somatic gene transfer will be taking most of this presentation and will be the highlight of this presentation. Let me just kind of stay as basic information with respect to how we deliver components of the Sleeping Beauty transposon system. Don't forget, we're talking about two components, a transposase component, which is a protein, and a transposon component, which is a nucleic acid. It's a DNA. We typically use a so-called two plasmid system. What it really means is that we mix together two plasmids together, and we deliver these two plasmids together into a target cell population. The two plasmids are exactly the ones that are required for transposition to take place. A transposon, an artificial transposon maintained on a plasmid containing a gene of interest, for example, a therapeutically relevant transgene that is flanked by the terminal inverted repeats of the transposon, and a separate plasmid, which is an expression vector that will express the transposase. We can also use a configuration of a one plasmid system. We can combine these two factors on a single plasmid. We can use, as a source of the transposase, not only an expression vector, we can use messenger RNA to express the transposase, as well as a recombinantly produced transposase polypeptide. This is something that I will cover towards the very end of this talk. Now, in the laboratory, we can use an entire array of transfection technologies, lipids, nanoparticles, liposomes. There's a vast array of technologies that are applicable to shuttle naked nucleic acids into cells. Electroporation is another technology that we typically use. And so you appreciate Sleeping Beauty or any other transposon, as I said, is a non-viral vector system in this particular context. This is great in certain aspects, and then I will highlight some of the advantages of this system, but there's also certain disadvantages that come with the non-viral gene vector. Namely, is that naked nucleic acids apparently don't have an active mechanism to be shuttled into target cells. So we always have to use some kind of a technology, transfection, electroporation, and other technologies to actually pump these reagents into cells. And that brings me to the last possibility that I wanted to highlight here. We can take advantage of the natural ability of certain viruses. So for example, the non-integrating viruses of a baculovirus or an integration deficient lentiviral vector or an adeno-associated virus to actually package the components of the transposon, taking advantage of the virus to actually shuttle the components very actively and very efficiently into the target cell population. And once the components, the nucleic acid components of the Sleeping Beauty vector system are inside the nucleus, then transposition will take place and the transposon components will kick in and allow stable genetic integration in that particular cell. So this is kind of the baseline, kind of the rough basics of how this transposon technology works and what are the different components of this. Now, let me give you a few examples for how we actually apply this, this vector system in two areas. The first area is germline gene transfer. What do we mean by this? What we typically mean by germline gene transfer or transgenic animals, it's another term that covers this area of research, is actually dependent on a fairly old technology, which is called pronuclear microinjection. Is a physical technique that uses a needle, a very fine glass needle, to actually deposit naked nucleic acids into a freshly fertilized oocyte. This was initially established for the mouse system. So this Sleeping Beauty application is practically speaking using this pronuclear microinjection technology. The only difference is that a gene of interest, in this particular case, a venous fluorescent marker gene, is now incorporated into a Sleeping Beauty vector, which is maintained in the form of a plasmid. And as I said, the transposase can be provided in the form of messenger RNA. So all what we have to do in such an experiment is to mix together two nucleic acids, a plasmid that contains this artificial transposon, and messenger RNA encoding the Sleeping Beauty transposase, and then micro-inject this mixture into fertilized zygotes, fertilized eggs. And then the activity, and this is shown here as just an example in the mouse system. The stable transgenic animals are fluorescent. Obviously, they incorporate their venous transgene very early on, and then every single cell of this mouse will be fluorescent. And the activity of this system is at a level that allows you to, practically speaking, derive, say, if you have 10 animals born out of micro-injected embryos, after embryo transfer, chances are that 5 of them, say 50% of them, will be stable transgenic founders like the one highlighted in this figure. So it's a very simple technology and quite efficient. I'd like to highlight on this particular slide one quite important and interesting observation. On the upper panel of this slide, you see transgenic mice that incorporate the venous fluorescence gene delivered by this particular technology. And what I really wanted to highlight in this panel is an increasing intensity of this fluorescence intensity signal as the numbers of the integrated transposon copies increase. So you can appreciate that an animal that has 3 copies or 4 copies of this integrated transgene develops a more intensive fluorescent signal than another animal that has only 1 copy. Very similar observation, and this is now the bottom part of this slide, transgenic pigs that have been generated by micro-injection of the Sleeping Beauty transposon components, delivering the very, very similar message, showing that transgenic pigs that contain 2 copies of the Sleeping Beauty transposon carrying this fluorescent marker gene do develop a more intensive fluorescent phenotype than another animal that has only 1 copy of the same transgene. The major message or the implication of this information or this observation is that apparently most of these transgene copies are expressed even following germline transmission, and apparently transgene silencing following Sleeping Beauty gene delivery is very minimal. So we can be more or less certain that if we end up with transgene integrations in a target cell or in a target organism, those integrated transgenes have a very, very high chance of being expressed long term. Now, this leads me to kind of the area of applications that I really wanted to highlight in this particular presentation. And that leads me to the area and the topic of cell engineering for gene therapy. Now, it could be that other speakers of this lecture series already touched upon a little bit ex vivo gene therapy and how this works typically in the context of clinical applications. Let me quickly go through this pipeline that is also geared towards the use of transposons a little bit. So ex vivo means that we manipulate cells that are recovered from a patient. So genetic manipulation takes place in a petri dish or in a flask. It doesn't take place in the body of the patient. In case we're using the Sleeping Beauty transposon systems, such cells, it can be a variety of different cell types, and this is something that I will show to you in the next couple of minutes by using a few examples. These cells are recovered. They undergo genetic manipulation by Sleeping Beauty transposon vectors. For primary human cell types, we typically use electroporation as a technology to shuttle these transposon vectors into the cells. And following a brief period of enrichment of genetically manipulated cells, one can obviously use animal models for assessing preclinical efficacy and safety aspects of this procedure. And of course, down the line, the major goal is to test this approach in clinical trials in human patients that would include infusion of this genetically manipulated cell population back into the same patient in case we're talking about an autologous application, and that is the donor and the recipient of the cell population is the exact same person. So let me now give you kind of three independent examples for clinically relevant applications. The very first application leads me to an eye disease, which is called age-related macular degeneration. And I had the privilege of being a partner of an European FP7 funded research consortium that has been aiming at providing a non-viral Sleeping Beauty-based gene therapy to tackle this particular disease, AMD. Just to give you kind of a feeling what AMD means, AMD is a neurodegenerative disorder affecting the vision of people, typically affecting the elderly population. And if you happen to have AMD, this would be something that you would experience in your field of vision, clearly a condition that has a significant impact on the quality of life of these patients. Now, let me just give you the background of AMD. So the pathology of AMD can be traced back to an imbalance of two signaling pathways at the retina of the eye, namely an angiogenic and an anti-angiogenic signaling pathway dedicated or controlled by two factors, VEGF and PDF. When there's an imbalance and an overrepresentation and upregulation of VEGF, this imbalance will lead to an angiogenic signal cascade, extensive blood vessel formation at the retina, and physical damage of the photoreceptors, leading to this particular pathology. The current treatment of AMD, one particular form of the AMD, I have to add, is an injection of anti-VEGF antibodies on a regular basis into directly into the eyes of the patients to kind of reinstate this imbalance. And another possibility, of course, is to reinstate this imbalance by overexpressing an anti-angiogenic factor, which is called PEDF. And that has been exactly the goal of this particular consortium, Target AMD. So the concept of this consortium has been that autologous cells from the eye can be recovered from the patients. This can be either recovered from the retinal pigment epithelial cells, or from the iris, which are called iris pigment epithelial cells. This is a classical ex vivo application. So again, these cells are taken into an electroporation device, electroporated by Sleeping Beauty transposon vectors. And the concept that is relying on having a therapeutic gene, that is the PEDF anti-angiogenic factor. And once this cell population is expressing that anti-angiogenic factor and transplanted or grafted onto the retina of the eye, following genetic manipulation, would release and as a little bioreactor would produce that factor in the local area of the eye, thereby inhibiting blood vessel formation. Just 2 points down here on this slide, a proof of concept that shows to you by applying a GFP marker in preclinical experiments that GFP expression is long-term in these ex vivo transfection experiments. It lasts for years. That's point number one. And point number two is that human PEDF apparently can be detected after long periods of time from very, very limited numbers of cells following Sleeping Beauty gene delivery. So that's kind of the concept of this consortium. And I also wanted to share one preclinical data slide video from colleagues Sandra Jonen and Gabriele Thumann, who have shown and published this very nice paper in which they quantified this area in a rat model, which is called neovascularization area. This is kind of an area that is related to blood vessel formation by staining for proliferating endothelial cells in animals that have not received a cell product, basically a Sleeping Beauty engineered a cell product releasing and producing PEDF, as opposed to an experimental group of animals that received transplanted cells producing PEDF. And the bottom line message here is that those animals that have received the cell transplant display a reduced area of neovascularization. This is also shown on the right-hand area of this slide, apparently providing us with some kind of preclinical feedback with respect to potential efficacy also in humans. This consortium has been in contact with the regulatory agencies and still collecting preclinical data for an approval of a clinical trial, which will be a really exciting application of this particular technology in this particular context of human disease. Now, the second area that I wanted to cover leads me to hematopoietic disorders. Now, unlike pigment epithelial cells and retinal epithelial cells, hematopoietic stem cell-based gene therapy is based on a stem cell compartment, hematopoietic stem cells, which have the capacity, the biological capacity to differentiate into many different cell types in the blood. So in other words, the very basic concept in hematopoietic stem cell gene therapy is that once you introduce foreign genetic material and a genetic engineering or a genetic modification in the stem cell compartment, and you allow these hematopoietic stem cells to differentiate into the various different cell types of the blood, this particular transgene construct will be inherited and will also be expressed, hopefully, in the progeny in the differentiated cell types of the blood system, which means that following a single genetic engineering step, one can potentially target certain pathologies that affect many different cell types of the blood system. So we wanted to kind of tailor Sleeping Beauty technology for genetic engineering of human hematopoietic stem cells, CD34 positive bone marrow derived cells. And for this, we applied a little trick in vectorology of Sleeping Beauty. This little trick is called a minicircle vector arrangement. Minicircle is kind of a smaller plasmid, is a derivative of a classical plasmid, but it's much, much smaller in that a mini plasmid doesn't contain, or minicircle doesn't contain bacterial origin of replication or an antibiotic resistant gene. plasmid backbone in general. It's practically speaking nothing else but a circular transgene construct. So we vectorized Sleeping Beauty components, both a transposon vector, fluorescently tagged transposon vector, as well as a transposase expression cassette expressing a hyperactive version of the transposase, which is called SB100X. And in this particular experiment, we also wanted to test the transposase messenger RNA as a source of the transposase factor. The very first experiment that we did was addressing potential toxicity. Now, this is really important. If you electroporate naked nucleic acids, most of all plasmid constructs, into human CD34 cells, it's a highly toxic procedure. This is highlighted here. You kill a large fraction of cells just by electroporating these cells with plasmids. But importantly, when we replace the classical plasmid arrangement with the minicircle vectors, as well as minicircles supplemented with messenger RNA encoding the transposase, we could rescue some of the viability in this electroporated cell population. So point number one, viability of cells, and this particular technology allows you to recover a larger fraction of your cells in a viable form. The second point that I wanted to highlight is the actual efficiency of stable gene delivery. This experiment highlights to you readouts based on venous fluorescence. These are experiments that we have been doing, or we've done in collaboration with our colleagues in Madrid, Juan, Bueren, and their colleagues there. The major message is that when we score venous fluorescent positive cells at a time point, namely 15 days following electroporation, this is a time point where you cannot read venous fluorescence in samples that have been electroporated in the absence of transposase. Okay, so in the absence of transposase, venous fluorescence declines because there's no active mechanism to potentiate gene integration. So that's the actual readout, the fraction of cells that are venous fluorescent at day 15. And here's the actual data. Those cells that have been manipulated by minicircles as well as messenger RNA for the transposase provide us with very significant numbers of stably engineered cells in CD34 population. This is also long-term. This particular panel of this picture shows to you that in the presence of transposase, venous fluorescence is detectable over long periods of time in roughly 50% of the cell population. And as I said, if transposase is not present, venous fluorescence declines to an undetectable level. So this was very important. And finally, this particular slide shows to you that at least a fraction of these genetically manipulated human hematopoietic stem and progenitor cells maintain their potential to be grafted into an immune deficient mouse, a recipient mouse. 3 months post-transplantation, human CD45 positive compartment is being read out here in these particular animals, providing us with proof of concept that at least a fraction of these cells do engraft. And finally, in this other panel, venous fluorescence is also measured in these animals following transplantation and engraftment in these animals, thereby providing us with the proof of concept that we have a technology for ex vivo manipulation of these human cells that allows us to generate decent numbers of viable genetically manipulated CD34 cells that contain Sleeping Beauty mediated gene integration, and at least a fraction of these cells can be engrafted into recipient animals. And the third example leads me to CAR-T engineering. Probably all of you or many of you are familiar with the chimeric antigen receptor engineering in the context of cancer therapy. There are 2 products out there on the market, both of them against CD19 positive human lymphomas and leukemia. And of course, the goal here a little bit is to also put Sleeping Beauty on the map. There has been a couple of clinical trials ongoing in the United States, pioneered by Lawrence Cooper in Texas. These clinical trials have delivered a very important message, namely that this procedure is safe and is effective in CD19 core engineered T cells, T lymphocytes, human T lymphocytes. And just wanted to highlight one particular practical feature of engineering these CAR T cells. Again, this is ex vivo genetic engineering, namely that in these particular clinical trials, CAR T cells have been maintained ex vivo for a longer period of time. And this is something that we wanted to kind of address with updated technology to kind of shorten the time frame that CAR T cells spend in a flask ex vivo. And this is how we did it. We teamed up with a group of Michael Hudacek in Würzburg here in Germany. We vectorized a CD19 core Sleeping Beauty as Minicircle. As I already mentioned to you in the context of hematopoietic stem cell, the same thing applies to the Sleeping Beauty hyperactive transposase. This allows us, this little trick in vector upgrade to Minicircle allows us to engineer human CD8 positive T lymphocytes very efficiently. Over 50 or 60% of these T lymphocytes do express CD19. This is also quantified here on this particular slide. And the in vivo validation of the data has been provided by a xenographed mouse model. Human CD19 positive lymphoma, also tagged with the luciferase transgene, is being grafted into an immunodeficient mouse. And then a week later, these CAR T cells, CD19 CAR T cells, are injected into these animals, leading to a complete eradication of this human cancer out of these experimental animals, thereby providing a preclinical proof of concept. Now, the news, kind of an update on a clinical trial that a European Horizon 2020 consortium is aiming to implement in the context of multiple myeloma. In this particular case, the CAR transgene doesn't target CD19 as a tumor-associated antigen. It's a novel CAR construct against the SLEMF7 tumor-associated antigen and is proposed to be a multi-center clinical trial in 4 European countries. And we're very excited because a clinical trial in Germany based on this application was approved not so long ago. And that gives us excitement with respect to a clinical outlook for this particular application. Finally, I'd like to address in maybe 2 minutes of my time potential safety aspects of Sleeping Beauty engineering. Perhaps you recognize this photo, Barbara McClintock, who discovered transposons in the maize on the basis of insertional mutagenesis, the potential of transposons to integrate into genes, thereby leading to different changes in the phenotype. So obviously this can apply to a clinical application. This is a certain risk factor that we have to address. In this particular context, there are 2 important points to consider: vector copy number and genomic integration profile. Vector copy number typically means how many integrated copies of the transposon do we have per genome. This is an experiment that we did in CD34 cells in bulk cell population. The average copy number of Sleeping Beauty is roughly 0.4, 0.5. And in clonally derived cell clones in these cells, the majority of cells do contain a single copy of integrated transposons. And number 2, the integration profile, we use next generation sequencing technology to sequence and map transposon integrations onto the human genome and compare that map to retroviral and lentiviral integration maps. Retroviruses have been known to preferentially target the 5' regulatory regions of genes, namely the transcriptional start sites, lentiviral vectors have been known to preferentially target transcription units. And Sleeping Beauty, although it has statistically significant preference for transcription units, displays significantly lower preference for gene sequences as opposed to the other two vector systems. This is a highlight or a zoom into the transcriptional start sites. Again, MLV kind of showing a peak at the promoter region. Sleeping Beauty in green is more or less like a fully random transposon integration profile, and the expression of genes, there is some certain association with the frequency of integration as a function of how strongly a particular gene is expressed, but this is the lowest association for Sleeping Beauty as opposed to the other two vector system. And finally, I mentioned that recombinant transposase can be used as a source in genetic engineering system. Why is this important? If you consider a plasmid vector that expresses transposase, it has to locate into the nucleus, transcription has to take place, messenger RNA has to translocate into the cytoplasm, translation takes place, transposase translocates back into the nucleus where transposition takes place. If you provide a recombinant transposase that is ready to go, then you circumvent this entire reaction and you allow very fast kinetics of transposition. So that's point number one. Point number two, the half-life of recombinant transposase is much shorter than the transposase when you measure the half-life of the transposase if you express it from an expression vector. This is again a safety aspect. The shorter half-life of the transposase, the shorter the period the transposase spends inside the cell, the more you elevate the potential safety of that application. And finally, vector copy numbers that are potentiated by the Sleeping Beauty transposase protein are lower than vector copy numbers by the actual expression vector. And this is my last slide through which I just wanted to kind of highlight a very unexpected phenomenon, and namely a self-penetrating phenotype of the Sleeping Beauty transposase. What this means, you can practically speaking just pipette recombinant transposase in the tissue culture medium. Cells will take up the transposase and potentiate transgene integration in the cells. On this particular slide, you see CAR T cells and the generation after enrichment of T lymphocytes expressing a CAR transgene that has been delivered by the recombinant transposase. And this also shows that the recombinant transposase is localized in the nucleus, even in non-stimulated, non-cycling human lymphocytes, thereby establishing this highly unexpected and very interesting result. Again, lower copy numbers by recombinant transposase, but very potent cell-killing activity of CD19 expressing human cells. That leads me to summarize this presentation. What I really wanted to show to you is that transposons are non-viral gene delivery vectors, very simple setup, typically plasmid-based for gene transfer. Sleeping Beauty is entering clinical trials in human gene therapy, and then highlighted to you a couple of very exciting applications. Sleeping Beauty transposon vectors combined with Minicircle technology allow very efficient, stable engineering of human T cells and hematopoietic stem cells. The insertion profile of Sleeping Beauty is more or less random in human cells, as opposed to classical lentiviral and retroviral vectors, and perhaps this unbiased integration profile contributes to an advantage of the safety profile of this vector. And finally, GMP manufacturing of non-viral vectors has a competitive economic advantage over viruses. Just wanted to highlight that if you calculate the cost of a Sleeping Beauty non-viral vector system on a GMP grade, on a per patient basis, it's only a fraction of the cost that you would typically need to come up with in the context of a classical viral-based system, which is an important consideration for clinical implementation. Finally, obviously, I'd like to acknowledge my direct colleagues at the Paul Ehrlich Institute here in Langen in Germany. Would like to acknowledge the wonderful collaboration, without going into deep details, with many of our colleagues across Europe and elsewhere. And finally, of course, I also wanted to highlight the different funding sources that have been at our disposal to help these investigations. And finally, and this is the last slide that I wanted to show. Well, first of all, thank you very much for your very kind attention. I will be very happy to answer questions following this presentation. So that's kind of the talk itself is closed by that. And I just wanted to kind of pop up an advertisement slide for next week's presentation. And I will leave that slide up so that you can read that, familiarize yourself with the date, with the speaker and the topic. Thank you very much. This was it from my side, and I will be looking forward to your questions. Thanks a lot.
Let me close to your presentation, which was indeed very clear in this very complex field. So there are several questions coming, and I will try to summarize one by one. We will have about 10 minutes for some discussion, right? So there is a question coming from Alberto Auricchio, and he asks, how large is the smallest transposase coding sequence that you can use?
Yes. So there are different versions of the sleeping beauty transposase. I just highlighted one particular version, which is called SB100X, which is kind of the state-of-the-art transposase that contains amino acid replacement mutations in the transposase coding region. It's not longer and not shorter than the first generation transposase, which leads me to the actual answer, which is roughly 1 kb. So the sleeping beauty transposase coding region is more or less 1 kb piece of open reading frame that you typically either drive in the context of an expression vector from a suitable promoter, followed by a polyadenylation signal, which is kind of a classical setup for a transgene cassette. And whenever we talk about a format of using a synthetic messenger RNA, in vitro transcribed messenger RNA, to provide the transposase source, it's about 1,000 nucleotide long messenger RNA that we have to supply at some point. Okay. Fairly small and fairly compact protein in the end.
Thank you. Now there are several questions from Robert Shore. I will start with one of them, and if there is time, I will continue with more questions from this researcher. So how can you control the destination of the integration? And also related with this, what prevents uncontrolled integration and so overexpression of the transgene? And additionally, I would add, how can you control the number of copies of the transposon that are integrated?
Excellent questions. Thank you very much for these questions. So the first question relates to control. I have to conclude, at present, we don't have a perfect control over transposition. I made a remark towards the very end of my talk when I brought up recombinant protein as a source of transposase in transfected cells, and kind of used that little panel on my slide to show to you that if we're introducing transposase in the form of the ready-to-go recombinant transposase protein, then following a few days, this transposase is gone from the cells. There's no prolonged presence of the transposase in cells as opposed to an expression vector. If we use electroporation and a classical plasmid or minicircle-based expression vector, transposase will hang around in these cells for about a week. And obviously, and I assume that's kind of the core of your question, during this period of time, when the transposase is still present in the cells, there's nothing to actually stop the transposition reaction. Now, as long as transposition would be limited to catalyze transposition out of our minicircles or out of our gene vectors in general into the genome, this would not be a terribly big problem. But there is a certain risk factor that is related to transposition of already integrated transposon. So basically a gene copy, a transgene copy that integrates into the genome somewhere, and then the transposase is still there, maybe for 2 or 3 days or even longer period of time, and this transposase would now use this genetically or genomically integrated transgene and hop it over to another place. And this is a certain risk factor. We cannot fully control it, but certainly, and that was the point in my presentation, the recombinant protein limits that time period when the transposase is available for transposition. So that's number one. Number two, maybe you also imply in your question a little bit whether the human genome contains other transposons that could be mobilized by Sleeping Beauty transposase. It's not the case. Sleeping Beauty transposon has been reconstructed genetically or molecular biologically reconstructed out of fish, and the human genome doesn't contain Sleeping Beauty-like transposons. So whenever we use a kind of an artificial transposon as a vector and then provide a Sleeping Beauty transposase, these two components can interact with each other, but there's no danger with respect to the human genome or the integrity of the human genome in a clinical application, because the transposase doesn't really recognize any genetic element in the human genome. And your final question was, how do we control vector copy number? Again, there is no absolutely precise control over copy number. I'm referring to viral technologies where people typically settle for a certain MOI, multiplicity of infection, which is kind of a ratio of infectious particles per cell in a given transduction experiment. We can also use that quasi MOI in a non-viral genetic engineering experiment, in that we can cut back on the amount of the transposon DNA as well as on the relative amount or concentration of the transposase protein for both of these components per cell population, and thereby driving reactions towards relatively low copy numbers. But so, you know, these kind of little tricks or experimental opportunities are certainly at our disposal, but a very precise control, whether we want to have just one copy or whether we want to have 2 copies or 3 copies, this we certainly do not have at the present time.
Understood. Thank you. Another question from Arjun Padmanabhan. With higher concentrations of the Sleeping Beauty, was there any toxicity observed in different cell types?
We do. Thank you very much, first of all, for the question. As I highlighted, especially in the context of hematopoietic stem cells, but I also have to say any primary human stem cell population reacts in a relatively negative way to electroporation by naked nucleic acids. Altogether, this is a toxic procedure. With respect to high levels of transposase concentration, we haven't really received any kind of link or association for toxicities with respect to transposase expression. So first, let me just give you one example. We can take messenger RNA, as I indicated to you in the context of hematopoietic stem cells, to serve as a source of the transposase. And we can pump micrograms of that messenger RNA into CD34 positive cells without actually observing significant toxicity on the cell, on that cell population. So that leads me to kind of conclude that the relative amounts of the transposase that expressed in the target cell population do not directly contribute to cell toxicity. It's mainly the transposon DNA or the DNA as a nucleic acid that is actually delivered to these cells, which is toxic, rather than RNA or the protein itself.
Okay, and now the last question, because we do not have any more time, from Karina Hadju. I was wondering what is the maximum DNA cargo for the Sleeping Beauty system?
Excellent. Thank you for your question. I was really looking forward to that question. Unlike viruses, Sleeping Beauty doesn't have like an upper limit of cargo. I have to point out that the longer transgenes transpose less efficiently. There's a negative relationship between size and transposition efficiency. This is clear. Longer transgenes do not support as efficient transposition as shorter transgenes, but there's certainly no threshold. The biggest transgene constructs that have been seen to transpose at reasonable efficiencies in human embryonic stem cells are bacterial artificial chromosomes, BACs, that are at a level of, say, 100 kilobase pair in size. So apparently, entire bacterial artificial chromosomes can be transposed by Sleeping Beauty components. They do so relatively inefficiently, but nevertheless, certainly there is no upper limit that we have been bumped into. So that's a very important question and very important message with respect to Transposon technology. Thank you again for that question.
Okay, Zoltan, thanks a lot. I'm very sorry because there are several other questions that I've not been able to raise, but most probably Zoltan will be able to directly answer to those questions personally to all of you.
Looking forward, thank you.
So thank you very much, Zoltan. Thank you very much to the audience, and I look forward to seeing many of you in the seminar that is coming in a couple of minutes, and once again next Wednesday at the same time. Bye-bye.
Bye.